Okay, so thanks. I'm Ken Bloom. I am, among many other things, I am the Holland Computing Center of the University of Nebraska representative on the OSG Council. I can't call it Open Science, the OSG Council, and also a former chair of the council. And I only have held those roles after David Swanson's death. He was in both of those positions at the time of his very unfortunate death in the car crash about 4 years ago. I came to Nebraska 19 years ago. David was one of my earliest collaborators there. We worked with other people to get the CMS Tier 2 Center going at Nebraska, and that was sort of Nebraska's entree into OSG, and David really took that and got it going very well. And that's why you see Nebraska on so many slides that you've been seeing this morning. A lot of wonderful things about working with David. Certainly a great collaborator, a great person. One thing that he did remarkably well, he was a great mentor, a great developer of talent, and certainly the people that he found and got involved in things, it turned out to be really excellent. And I'm just to name two people who I've seen in the room today: Brian Balkman, Derek Weitzel, right, who would not be working on OSG had it not been for David. And so certainly in recognition of everything he did as a mentor, the OSG Council did want to establish an award in his honor and his memory that is for students. And this, the goal to give it to students who've been participants in the OSG school, the OSG user school, and gone on to do really great things in super users, not the root kind of super users, but very, very, very successful users of OSG resources, and gone on to do some good science with that. And the concept that we had, of course, was this was coming together in fall 2019, and the next OSG All Hands was going to be in March 2020. And we were going to invite David Widow-Randa to come to the first, for the first awarding of these prizes at the OSG All Hands in March 2020, which turned out to be the very first thing that got canceled off my schedule as the pandemic got going. And we've had several virtual OSG meetings since then. This is the first in-person one, and so finally, very glad that we could bring Rhonda here today to present the awards. And we've also brought back winners of the previous awards who've been able to come to the meeting today, and Rhonda will be introducing this year's awardees. So I will stop there and turn over to Frank for a moment. I guess you could use that one at this point.
So Ken pretty much said it all. Let me just make a couple of personal remarks. David was very special in two seemingly opposite ways. He was very understated and very laid back, very low key, and yet had a very influential vision of how and commitment to the OSG vision. And I said already some of these things earlier today, and he helped influence where we're going, helped make this strong push towards the national CI vision that I talked about earlier today, and was always sort of in my ear of what could we do here to grow this? What could we do there to grow this? And it's very gratifying to see that this is actually now happening. When we founded OSG, we thought, oh gee, wouldn't it be cool? We do this thing for that large experiment, but wouldn't it be cool if it also had a huge impact on the nation at large? And that's having a huge impact on the nation at large, very broadly across lots and lots of campuses. I think we've accomplished now in a sense, because 142 institutions is unique. I would think there's very little even globally that has that kind of profile across all of science. And that sort of was core to David's vision. And it's very nice to see that this has happened. it's just too bad that he wasn't part of it in the last few years and has to watch this now from wherever he is currently. And anyway, I wanted to honor David here at the first time that we're meeting in person to do this award. And I'll leave it at that. Thank you very much.
Okay, so as Ken mentioned, the first giving of this award had to be changed slightly because of the pandemic. So the recipients to this point have presented at virtual all hand meetings, but we wanted to give them the chance to come in person, to present their work in person. And so we have 4 previous recipients that we're going to give short lightning talks for each of them. I am going to be standing in for the first 2 who couldn't make it, but have provided slides and a script for me to talk about, which is good because I don't actually know anything about taxonomic classification. And so I'll be doing the first 2, and then we'll be inviting up some of our previous recipients to talk about their own work before we present certificates to both them and the recipients for this year. So let me get this in the right order. So one of our 2020 awardees, Zhonggang or John Lee, is in the space of genomics, and his work involved the kind of big picture science was we can understand how individual genes express themselves and turn into a certain phenotype. So this gene causes this, observable effect, but what we don't always understand is the exact physical mechanism for that effect. Why does A lead to B? And so that was the type of work that he was doing. And so this idea of system genomics. And so he gave 2 examples in his all hands presentation about his work that basically both involved needing to calculate lots of relationships. So the figures on the left are from a project where he had like 35 gene co-expression data sets, and he was running models on them, and there were like 75 different parameters for each of the models. There was something like 75 times 35 times something number of jobs that he needed to run, that on one computer would have taken something like 2 or 3 years, and he was able to do in something like a week, using resources at the Center for High Throughput Computing and on the OS pool. And on the right, another project, similar kind of idea, where he had a data set with a lot of SNPs of data, and was doing looking at gene interactions. So essentially, thank you for this many SNPs, a big matrix where you want to calculate the interaction of each pair. That was another kind of thing that would have taken 3, 4, 5 years if you had to run all of those little comparisons on one computer. And instead he was able to do it in hours to maybe like a week, running on the OS pool. So that's the work that he was doing, and the impact of high throughput computing on his research. From 2021, our recipient, Nicholas Cooley, who's at the University of Pittsburgh. So here I have a full script. Their work on the OSG typically falls into 2 categories. The first is benchmarking and testing experiments that facilitate construction, evaluation, and refinement of tools. So this lab does a lot of developing kind of informatics tools for genomic data. Or there are other categories that the OS pool helps them facilitate database scale testing of the effects that methods can have on how they interpret biological data. So the plot on the left, assuming that's ABCD there, is from a paper where they expanded a taxonomic classification tool to flexibly perform functional classification. So that's a way of the thing that's being classified is genes. This particular plot was chosen because it distills the work of the paper down pretty neatly into a series of quantifiable error rates, but also represents exhaustive cross-validation that was enabled by the OS pool and would likely not have been even attempted without the OS pool. This particular plot is usually referred to as an OCMC plot and shows curves for how classification errors trend against the two axes as their confidences are scaled. Additionally, the utility of reduced amino acid alphabets was explored in this paper. So it's a way of expressing amino acids in a more simplified form. They were able to search that in a more logical manner to find alphabets that improve classifier performance. With 5.1 trillion possible reduced alphabets, a DAG search facilitated by OSG tools allowed Nick and company to search a narrower space more quickly and identify useful alphabets for their work. The second plot on the right is work in progress from a paper that should be submitted very soon, seriously, exploring how different choices of sequencing technology, assembler, and sequenced organism affect reported pseudogene counts in sequence assemblies, reassemblies of raw SRA reads, and assembly of simulated reads. So all sorts of genetic data processing. This particular plot illustrates that under uniform quality control conditions, different assemblers, so different assembling codes, produce assemblies with different sets of pseudogenes. Okay. And to be able to do this at a large scale, to kind of try all these different combinations and iterations of these tools and data would not be possible without the OS pool. And there's even more, but it didn't fit on the slide. So that's presentations from or a kind of an update from our 2021 recipient, Nick Cooley. And now I'm going to bring up Anirvan Shukla and Connor Natzke to share about their work from the past couple of years. You're up first.
Hi, I'm Anirvan Shukla from the University of Hawaii at Manoa. And my association with the Open Science Grid started in 2016 when I attended the user school. And back then we were looking at the production of certain kinds of antimatter particles. Deuterons are matter and their corresponding antimatter particles are antideuterons. The Na61 experiment at CERN, to talk about how kind of rare these particles are, we had recorded approximately 100 million proton-proton collisions, and we were expecting to see maybe 100 to 1,000 deuterons, maybe 10 to 50 antideuterons. So sort of a needle in a haystack. My group was also working on the AMS experiment on the International Space Station, which was sort of gobbling up these cosmic rays that show up at Earth. And we have about 10 years of these cosmic ray data, and we have collected about 100 billion of these charged particles that we see. And we have been able to identify about 20 anti-helium candidates, which are extraordinarily rare. And I'm calling them candidates because we are not 100% sure that they are anti-helium. And so the rate is about 1 or 2 such particles a year. So it's very hard to identify them correctly. And also we have to separate the signal, which could be some exotic process or maybe dark matter from the regular astrophysical background. And the uncertainties in these are very high. And so the challenge was to separate them. So one of the things that we then were doing from 2016 to 2018 were looking at these particles, developing different processes. But then we realized that to really simulate how anti-helium propagates in our galaxy, we would need to conduct this very large-scale simulation, which was not possible with the resources we had in our own research group, which had a small cluster, maybe about 48 cores, and also from the University of Hawaii, which has a supercomputer called Mana, which had about 2,000 cores back then, and it would have taken an enormous amount of time to do that. So then I got back to the Open Science Grid, and one day I started experimenting, trying out some of the tools that I was developing for our cluster on the University of Hawaii server. And This needed about 100 trillion proton collisions to be simulated on the open science grid. That needed 68 million core hours, which is about 8,000 CPU years. And the data output that was produced in the simulation was about 100 million gigabytes. And obviously there's no way to pipeline it, to store it. So we had to come up with quote unquote smart techniques to reduce the size to maybe 1 million gigabytes, which was still not, it's easy to handle, but we were able to transfer that to our University of Hawaii server to do post-analysis after this pre-analysis. And then finally, if you look at the published results and the final data that we have is now finally condensed down to only 10 gigabytes. And so that's pretty neat. And the impact was that with this simulation, we could find the first cosmic anti-helium background using physics models. Before that, people were multiplying numbers and scaling these things without using actual physics. And we were able to use the Open Science Grid resources for 3 publications, 2 publications and a review publications, more than 100 citations. And it formed about a third of my PhD thesis. So I'm very glad and thankful. In future, our group is also involved in this NASA experiment called GAPS, which is going to be flying in 2024. That's not a hashtag, that's fingers crossed. It's going to be a large balloon experiment, about 2.5 tons. It's going to fly about 40 kilometers above the South Pole. And it's also going to be looking at these cosmic rays that kind of get funneled in due to the Earth's magnetic field. But there's a lot of simulations that we need to do, lots of new things that we have to deal with when you're within the Earth's atmosphere, because there could be backscattering, and we have to do a lot of the Monte Carlo calculations and sensitivity calculations. So we hope to be using the Open Science Grid resources even more in future. Thank you.
There you go.
All right, so I thought I had a lot of data, but I have a pittance compared to that. For anyone I haven't met, my name is Connor Natzke. I'm a PhD student at Colorado School of Mines, and I've actually spent the last few years stationed up at Triumph in Vancouver, British Columbia. Triumph is a particle accelerator lab, and I work in the low energy nuclear physics side, which essentially just boils down to measuring light very, very well. So just, does everyone know what a gamma ray is? Or at least heard of it? You know, it's the thing that makes you a Hulk if you're not careful with it. But in essence, all a gamma ray is you have 2 different, image at the top, you have 2 different energy levels inside a nucleus. So just thought of this stupid little joke because I was standing there. It's like, you know, you're a little over caffeinated in the morning and you need to kind of settle back down. The nucleus experiences a very similar thing, except to de-excite, it goes through all these different decay modes. It can release an electron, it can release a proton, it can split into a few different nuclei, or it releases high energy light, which is what we call a photon. And if it's sufficiently high energy, we call it a gamma ray. And normally, when this process, the nucleus undergoes a decay it releases 1 photon for that de-excitation to go down an energy level. In rare cases, it emits 2 simultaneous photons that, you know, couple together and interact in such a way that they act, you know, they carry away the same energy and the same angular momentum as 1. This is a rare process and it's hard to measure. So you need a big array. And generally, these big detector arrays here at the bottom are inefficient because they use basically a piece of 99.9 followed by 9 more 9s % pure germanium. The light interacts inside the germanium, knocks a few electrons around. You collect those electrons, and the number of electrons you collect equal the energy of the decay. So it's not a very efficient process. You know, light, most things are transparent to photons. So we collect, you know, you have a process that doesn't happen very often, and you have an array that doesn't measure very many events. and you kind of run into this cascading problem of not being able to see much. You eventually, you count for hours and hours and hours. I had about 10 terabytes of data or so, again, a fraction of what we just saw before. But you eventually do all this analysis, and at some point you have to make corrections because the system is not perfect. Your detectors are not infinitely small and infinitely efficient. So you have to model all of these processes in software, run simulations, and eventually, if you run enough simulations, you start to understand what the efficiency of your array is and what correction factors you need to add to anything you measure. So that's what this plot essentially is. It's deceptively simple. On the y-axis, it's really just a measure of efficiency. So for every 1,000 gamma rays that are emitted by my source, how many do I actually see? And then on the bottom, it's a function of energy. So the higher in energy you go, the more transparent matter is to gamma rays. So you see less and less and less. Again, this is a rare process in a very complicated array. So to generate each one of those points takes about 300 days of compute time. And that's assuming I do it right the first time, which, I'm a physicist masquerading as a programmer, so likely not. So really what my current OSG usage has been is using Monte Carlo simulations. And since it's distributed high throughput computing, these scale really well to just, I use up all the resources I can possibly get, since all the events are stochastically independent. I just add them together at the end. And just building out these efficiency curves for a few different processes and a few different orientations of the array. So without the OSG, let's see what there's, I think 7 or 8 points on there. That'd be 7 or 8 years. And I don't know, I don't want to do my PhD that much longer. So the OSG has really helped with that. And it also really helped develop a sense of how to break up large problems into much smaller chunks so that they are, actually usable on a human lifespan. So thank you again. And if you have any questions or want to talk further, just feel free to find me.